Proteases break peptide bonds. In the lab, it is often necessary to measure and/or compare the activity of proteases. Sigma's Universal Protease Activity Assay may be used as a standardized procedure to determine the activity of proteases, which is what we do during our quality control procedures. In this assay, casein acts as a substrate. When the protease we are testing digests casein, the amino acid tyrosine is liberated along with other amino acids and peptide fragments. Free tyrosine then reacts with Folin and ceocaldius phenol, or Folin's reagent, to produce a blue-colored chromophore, which is quantifiable and measured as an absorbance value on the spectrophotometer. The more tyrosine that is released from casein, the more the chromophores are generated and the stronger the activity of the protease. Absorbance values generated by the activity of the protease are compared to a standard curve, which is generated by reacting known quantities of tyrosine with the FC reagent to correlate changes in absorbance with the amount of tyrosine in micromoles. From the standard curve, the activity of protease samples can be determined in terms of units, which is the amount of micromoles of tyrosine equivalents released from casein per minute.
Hello, my name is Carrie Cupp-Enyard, and I'm a scientist at our Sigma-Aldrich DeKalb facility here in St. Louis, Missouri. Today, I will show you a universally applicable way to test protease activity. At Sigma-Aldrich, we mainly use this assay in our quality control area to ensure that our protease has sufficient activity before we ship it directly to you at your lab.
Before beginning the assay, we need to make sure that the following reagents are correctly prepared: a 50 millimolar potassium phosphate buffer,